This week in Virology, episode number 99, recorded on September 13th, 2010. Hey everybody, I'm Vincent Racaniello, and this is TWiV. And today we have a special TWiV. We're coming from Boston, Massachusetts, at the 50th Interscience Conference on Antimicrobial Agents and Chemotherapy. This is a huge meeting that attracts over 10,000 physicians, scientists, and other healthcare workers to talk about infectious diseases. And all your favorite microbes are here: viruses, bacteria, fungi, even eukaryotic parasites. So I'm ready to go to this meeting. I've got my abstract book, which tells me exactly what's going on. I've got my nice little bag here from the meeting. So let's go inside and check out what a really big science conference is, and maybe talk to some of the scientists who are talking here today. Inside the Boston Convention Center, and the first thing you can see is that it's a brand new building. It's part of a convention area that was built at the waterfront on Boston, includes many hotels and easy access to transportation. For a big meeting, it's the place to be. Now, as we enter this open foyer here, you can see on either side an area called East Central, and that's where you can check your email if you haven't brought your laptop. And you can see on both sides of me there are about 40 kiosks set up where you can do that. Over at the other end of this foyer is the headquarters of ASM Press, and let's check that out and see what books they have there. So we're standing in front of the booth for ASM Press, and this has a very prominent position in the lobby because ASM, the American Society for Microbiology, is the organization that sponsors this meeting, ICAAC. And this is the press that published my virology textbook. So let's go inside and see if they're actually selling it. Looks like we've found it. Here they are, volume 1 and 2, Principles of Virology. Awfully large stack here, though. I don't know if they've sold any. We'll have to find out. But anyway, ASM Press publishes a lot of very interesting books on microbiology. You can see the shelves are loaded with them. There are other virology texts, molecular biology, all aspects of microbiology. Let's find out if they've sold any of these. Can I ask you a question?
Sure.
There's a textbook over there called Principles of Virology. You've sold any at this meeting?
We have, yeah.
Okay, I'd like to know because I'm a co-author on that book. Well, keep pushing it.
In general, I mean, there's a lot of interest in it always, so yeah.
Thanks for talking. I'm talking with Professor Derek Schmidt from the University of Cambridge in the UK, an old friend of mine. Hey, Derek.
Hey, Vincent, how are you?
Finally caught up with you after trying for 2 years to get you on TWiV.
It's been a while.
Derek spoke today, just in a past session, about a really neat technique he developed called antigenic cartography. I want to explore that a little with you today, because our listeners would love to hear it. But it's based on getting a lot of samples. And
that's right.
I think you mentioned how the World Health Organization is involved in that. How does that work?
Yeah, when the influenza vaccine was first made, it worked great for a few years and then it stopped working, and the reason is because, and this was in the 40s and the 50s, the virus had evolved. So the strain of flu that was used in the vaccine was updated, and then a couple of years later, the vaccine stopped working well again. And about 50 years ago now, the World Health Organization instigated something called the Global Influenza Surveillance Network. Mm-hmm. And this is thousands of people around the world who take samples from people who have flu. Those samples are sent to one of over 100 national influenza laboratories around the world, where they're tested to see what type of flu they are and sort of details about how the viruses have evolved. And those samples are also sent to one of 4 international collaborating centers on flu, also WHO centers, either in Australia, Japan, the UK, or the US, and there they're analyzed in great detail. Depending on how much flu there is in a particular season, there might be anything between 10 and 20,000 strains that are analyzed this way each year.
Globally?
Globally, that's right.
Is it all influenza A or also influenza B?
Also influenza B, that's right.
And there's a committee that oversees the results, and you're part of that committee?
Yes,
is
that's
that
right.
correct?
Yeah, the committee that selects the strains of flu that go in the influenza vaccine. So this committee meets twice a year to decide whether or not to update the vaccine strains.
Are they always right?
They always do a really good job. And the times when, but there is a risk because that committee meets about a year in advance of when there will actually be a flu season. It has to meet sufficiently in advance because there's now about 300 million doses of vaccine that are produced, and that vaccine has to be made and tested, distributed, people vaccinated.
So a lot rides on this global network.
It's a hugely important decision, and yeah. But what can happen is occasionally the virus evolves during that period from when the decision is made until when the vaccine is used. There are 3 different strains of flu that are in the vaccine: one against H1, one against H3, and one against B. Occasionally, one of those components might not be a perfect antigenic match. Even when it's not a perfect antigenic match, it still protects people, vulnerable people, against hospitalization, is what the data says.
So what data do you use to try and predict whether a particular strain is going to be the one that you should immunize against?
Yeah, so there's a number of different pieces of data that are used. There's genetic data, there's epidemiological data, there's human serology data. The most critical data are these detailed so-called antigenic analyses of the data. That this indicates, this is the data that tells us how far the strains of flu have evolved in the sense of how our immune systems will see that difference. So this is the critical thing for deciding what strains of flu should be in the vaccine.
So these involve hemagglutination inhibition, or HI tests?
That's exactly
as we call
right.
them.
That's
so
exactly
we
right.
have talked about this on TWiV before, so some of our listeners will be familiar with it.
Yep.
So essentially, if you find a new strain that's prevalent in a particular region, that's distant enough from the previous season's virus, you decide to use that. How much different does it have to be in terms of this HI test?
Yeah. So it can't just be different in one particular region. It has to be different, and it looks like it's going to spread around the world, because what we're really trying to do is we're trying to estimate what strains are going to be circulating about a year from when the choice is made. And as I say, this is typically done very well and very accurately.
How do you know if it's going to spread, though?
A lot of historical information about how things have spread before. In fact, there was a very nice study that was published in 2008 in Science with people throughout the WHO Collaborating Center Network, where they found out that for the H3 viruses, that every year these viruses, they are continually circulating in East and Southeast Asia, and every year the viruses that we get in the rest of the world are the viruses that are circulating in East and Southeast Asia. So this is a particularly important place to look every year for the H3 viruses.
So your technique of antigenic cartography taps into these type of data, the serology that we're talking about. So can you explain it to us?
Yeah. So these data have been studied for many years in a tabular form. Tables of data of viruses that have been tested, as I say, thousands of viruses that have been tested against, somewhere typically between about 8 and 16 different antisera. These data in a good laboratory, they are very repeatable data, and they are highly quantitative data. However, they're difficult to understand for anybody but just a very few number of experts in the world that somehow seem to be able to make these things we call antigenic maps in their heads from looking at the tabular data. What antigenic cartography does, it takes that tabular data and does some mathematical and computational techniques. It's absolutely not rocket science, it's pretty straightforward, and converts it into something we call an antigenic map, which is a visual representation of that data. It's a similar sort of thing if you look, for example, in the back of a road atlas. There'll be typically see a table there that tells you how many miles or how many kilometers it is between different cities. It's very easy to imagine that if you already have a map of, let's say, the United States, you can easily generate that distance matrix, what a mathematician would call that table of miles between cities. It turns out that it's a pretty easy thing to do computationally and mathematically, to take that distance matrix and reconstruct the map of the US, or more specifically to reconstruct the relative positions of the cities. What antigenic cartography does is it takes an HI table, this table of data for how antibodies react with different viruses, and it constructs something we call an antigenic map. which looks a little bit like a geographic map, but instead of having all the things we usually see in a geographic map, what it has are antigens, the different strains, in this case of influenza virus, and antisera, the antisera that have been used, that they've been measured against. And we can see, depending on how the strains are clustered, how related those strains are to each other. So the closer that strains are to each other in an antigenic map, the more similar they are to each other antigenically, and thus how well a vaccine strain that is in the center of a cluster of viruses will be protective against those viruses.
So in your talk today, you showed some of these maps.
Yes.
And you showed in particular a lot of data for the H3N2 viruses.
That's right.
Which arose in 1968.
That's right.
And are still circulating.
They're still circulating today.
And you showed a cluster of spots of one color.
Yes.
Red spots and then purple spots quite a distance away, then green spots.
Yes.
And you called them clusters.
That's right.
So what does that mean?
Well, it turns out that influenza viruses, the H3 viruses that are circulating globally, typically tend to be very related to each other. So there are thousands of strains that are analyzed in any particular year. Somewhere between 5 and 20% of the world's population gets flu, which means that there's maybe something like 1 billion people who get infected with flu every year. All of the data indicate that these strains tend to all be pretty similar to each other. They tend to be in a cluster antigenically. It's really pretty fortunate that they are, because the fact that they are means that we can choose one vaccine strain, a representative strain of that cluster, and it will protect against all these people.
So the viruses within a cluster are all protected by a single vaccine.
That's right.
Okay.
That's right. And then the way that the virus happens to evolve is it typically will form a new cluster of viruses that's some distance away from the previous cluster, and the old cluster will go extinct. And this has happened now something like 14 or 15 times since H3 entered the human population in 1968. And each time there's one of those cluster transitions, it's when the vaccine needs to be updated.
Now, you obtain these data after the fact, but could you predict the next cluster that's going to arise?
Yeah, well, as you say, we've known each other for a while, and so you know that the primary thing that we do in our research in Cambridge, and also with colleagues at Erasmus Medical Center in the Netherlands and other people throughout the world, is to try to see how predictable this evolution might be. You know, and of course, it's never going to be, well, it's unlikely to be predictable many years down the road. But if we could get that, if we could sort of move forward that prediction horizon to around about 1 year, it would have tremendous impact for public health reasons, because instead of doing these very good estimates of which strains are going to be circulating a year later, we would have a much better idea of what was actually going to happen. It's unlikely to ever be perfect in the same way as with weather forecasting. You know, today the weather forecast is pretty good sort of 2 or 3 days into the future in most parts of the world, but it's never going to be 100% right. But if you can, well, you know what I mean.
Sure. Yeah.
So there's a couple of things. One's the prediction horizon, in the sense of about how far in advance one can predict, and then the other issue is how accurately.
Right.
We don't know yet whether or not flu evolution, or the evolution of any pathogen, is going to be predictable at all. We have some indications that it is, which is why we're putting a lot of effort into it, but until we actually figure it out, we won't know one way or the other.
Can you apply the technique to other viruses as well, besides flu?
Yes. Already published is the application to lyssaviruses. Rabies virus is a lyssavirus, to EV71, and to some more influenza viruses, CTL epitope, the T-cell response to influenza virus. There's also work going on malaria. There's work about to start on dengue and HIV. So the method is a very general method, nothing specific at all about flu. Our strategy is a lot of deep investigations on flu, because of all the antigenically variable pathogens, it's really sort of the most simple of them to try to hopefully, thoroughly thresh out the methods there, potentially thresh out some underlying principles there that can be applied in these more complex situations.
I would think for HIV it would be hard, because the variation occurs within an individual.
That's
and
right.
You would have to have multiple samples of antibodies.
Yeah, that's absolutely right.
But it
absolutely
would
right.
be interesting to look at that as well. So why did you develop this approach? Was it to be able to predict or just to see what had happened already?
Well, I say that necessity is the mother of invention. And in the late 1990s, I and colleagues were working on the repeated vaccination, the effect of repeatedly vaccinating individuals against influenza.
Right.
And we came up with with an underlying mechanism that we felt was applying when people are being vaccinated repeatedly for some of the variation in efficacy that one is seen when that happens. And we wanted to test this method. We tested it on all the existing data. It looks like this particular mechanism held. So it was really time to think about applying this method in practice. And one of the things that we needed was to have high-resolution information about how the virus is evolving. And so the data that we needed to look at, this phenotypic data, the antigenic data, was in these HI tables. And when one looks at these HI tables, as I say, they're very quantitative, but not well understood by almost everybody because of these paradoxes or irregularities in the data that we talked about before. So Ron Fouché, Alan Lapides, and I set about trying to understand what these paradoxes were about, and in order to be able to have this high-resolution quantitative assessment of these antigenic differences. And then it just grew into this monster where we found out we could do all these other things with it as well.
Is there a place where people can go to look at your maps and find out about them?
There is. If they go to www.antigenic-cartography.org, there's information there about antigenic cartography, and we'd be very happy to see them.
Excellent. I want to ask you one more question. I know you have to catch a plane to go back home.
Yep.
One more question. What's going on with the pandemic H1N1 virus in terms of antigenic cartography?
Well, the virus is so far not evolving antigenically from where it's been since the pandemic started, of course, which is great news. It is, however, expected that it will evolve. It's going to turn, almost certainly will turn into a regular seasonal influenza virus, and like we see with all other seasonal influenza viruses in humans, there's going to come a time when the current antigenic cluster evolves into the next cluster. It's not something that the WHO network is seeing yet, but everybody's on the watch for it, and as I say, every 6 months there's a new vaccine decision that is made for whether the strain should be updated. In fact, in the end of September, which is just in a week or so from now, the WHO, this committee will meet to decide what the vaccine strain should be for the Southern Hemisphere winter. Of course, that's out of phase with the Northern Hemisphere winter. So that choice is made in September every year for the people in the Southern Hemisphere to be vaccinated. For what would be
our summer next year.
For what would be our summer, that's exactly right.
And given historical data, the change happens every 1 to 3 years, roughly? Is that right?
For H3, it's something like that. It can be longer. On average for H3 since 1968, it was 4.5 years, although some antigenic clusters, will last for just a single year.
What about the H1N1s that circulated from 77 till last year? What was the periodicity?
A little bit slower evolution, antigenic evolution in the H1s. Yeah.
Very good. Well, I think you're heading off to that meeting, the WHO meeting very soon.
That's right.
Great. Sounds exciting. Thanks for talking with us.
My pleasure.
Appreciate
Good to talk to you,
You too,
Vince.
Derek. Take
Bye.
care. Good trip home.
Thanks.
We're standing in front of the exhibitors floor here at the meeting, and you can see it's a huge space. And this is where a variety of vendors talk about what they sell. And these range from companies selling printed information, books, CDs, laboratory equipment, antibiotics, and other materials that could be used in chemotherapy of infections. And as you can see, it's always a busy place. There are many booths and many people always interacting with the people at the booths. So let's go down and see if we can talk to some of them and find out exactly what they do. And there is a ton of stuff here. And in the back are the posters as well, which we'll go take a look at also. I'm always amazed at every meeting this gets bigger and bigger. Even with the internet, it still seems important to have FaceTime with people. I'm speaking with Michelle and Maureen from Vertex Medical Information. Hi.
Hi, how are you?
So tell us, what does Vertex do?
Vertex is a pharma company in Cambridge, Massachusetts. We have an extensive pipeline, but we are here today to answer questions about our protease inhibitor for hepatitis C.
And do you give out information in printed form, CD, electronically, or all of the above?
All of the above. We have disease state information about hepatitis C. We offer several slide decks that we're handing out to interested practitioners. And should anyone have questions about our pipeline, we're happy to provide written information as well.
So do you think coming to a meeting like this is useful for informing the people who use your products?
Absolutely. We don't have a product on the market at this point in time, but there has been a lot of interest in our company and our pipeline and what we anticipate. We just finished our phase 3 program, so we're in a pre-launch period, and so there's been a lot of interest about our drug and when it's coming to market.
Great, thanks for talking to us.
Thank you.
You're welcome. We're in the poster area, where there are rows and rows of these boards where people put up their results and have people come and talk to them. And I'm talking with Michael Schmidt in front of his poster right now. He's going to tell us a little bit about what we have. You can see it's a single sheet of paper on which there is information as well as photographs. And Michael, what do you have particular on this poster?
Well, we have an exciting story from the perspective of acquisition of microbes from the environment. Staphylococcal infections are the most prevalent in hospitals, and so we asked a really simple question about where bacteria are on common objects, and this particular object that we looked at is the common phlebotomy chair or the infusion chair, in which patients would actually sit and actually have an infusion of antibiotics or dialysis, or in this particular case have their blood drawn. So we asked a couple of simple questions. We said, on plastic surfaces, what is the bacterial load? And what we learned is that the microbes are at about 1,290 colony-forming units per 100 centimeters squared. There's been published reports in the literature that suggest anything above 500 colony-forming units represents a risk for the acquisition of the microbe and then ultimately an infection caused by that microbe. And so, as you can see, the tray table that the patient would rest their arms or read a magazine as they were getting the infusion does represent a substantial risk to them. And similarly, the arms, I don't know about you, but when they stick me to draw blood, I'll actually hold on. And so you can see that, again, they have a similar concentration of microbe than does the over tray table. So it's an old observation that metallic copper is antimicrobial. The ancient Phoenicians actually sharpened their swords, and the shavings from the sharpening they would actually place over the wounds they acquired in battle. And they learned empirically that when the copper shavings hit their wounds, they wouldn't lose their limbs to gangrene. Whereas if they didn't, the limbs would actually be lost, and that's why surgeons had to be quick at cutting off limbs. So antimicrobial copper is an old observation, and so we asked a simple question: if we put antimicrobial copper trays or arms, what's the level of risk going to be to that particular patient? And as you can see, 1290 down to 150, it drops it by well over an order of magnitude, and more importantly, it's below that magic red line of 500 colony-forming units per 100 centimeters. And when we looked at it, because bacteria are everywhere, they're randomly assorted in the environment, and when we asked the question, at that 500 number, how many were above 500 on copper? Only 28% out of all the samples that we looked at. The majority, 38%, were 0, and between 1 and 500, as you can see, there were only 33%. So copper is really doing a number, and when we computed the risk, if you sit in this simply copper-covered chair, you had a 17-fold lower risk of exposure than if you sat in a standard chair. So we're looking at this from the perspective of reducing the likelihood of an infection that you get from just normal health care. And the dollars and cents of it are, the CDC did a study that showed that the average hospital infection adds 208% to your bill. So while in the United States we really don't worry about money in healthcare, overseas, the Europeans, 208% is real money because that's their tax dollars because they have state-funded healthcare. And so the Europeans are very much interested in incorporating common touch surfaces with antimicrobial surfaces like copper to limit the spread of infectious agents as well as lowering their cost.
So what you just told us would be a typical presentation to a visitor to your poster at this meeting. Would that be correct? So did you have a lot of visitors at this meeting?
We had a large number of visitors from the U.S. as well as many of the Europeans. The Europeans were very much intrigued by the percentage in which we saw no colonies, and the Americans were by here being very curious, asking, how do we convince our administrators to do this?
So what is the real value of a poster as opposed to giving an oral presentation where you could reach 1,000 people, where here maybe 50 or 100 in one time?
The value of posters is the opportunity to interact with colleagues, to chat with them about their problems, some of their solutions. And one of the neat things about posters is they group like science together. So you meet your colleagues and you learn about things that they're doing that may help you with your work. And so this one-on-one interaction at the posters is really what posters are all about, the interplay of science. And whereas a platform talk reaches a larger audience, they may not actually be the ones who can help you affect your science or similarly help them affect their science.
So it's really in the end the one-on-one, the FaceTime that's important. And even with all the technology we have, a piece of paper on the wall still does a lot. Thanks for talking with us, Michael.
Thanks, Vince.
Now I ought to point out that this is a sheet of paper on which everything is printed. So this comes out of a high resolution, big size printer. Now when I was a youngster, we didn't have this. We used to print sheets of paper, individual 8x11 sheets of paper, and tack them to the wall. Our photographs, you probably remember that time as well. But things have changed, and now you can print this out. And in fact, right here at the meeting, there is a facility where you can print out your posters, and I'd like to show you that now. We're here with Jennifer at iPosters, which is a kiosk of 6 stations. What do you exactly do at iPosters?
Well, I was really interested in getting a poster so I could read later on this evening, and the abstracts are in the abstract book, and it's the first time that not a lot of the posters are available for use when you look at the abstracts. So I came to the iPoster booth and got some help from my colleague over here, and it's very easy. All you have to do is put in the abstract number and search, and the poster pops up, and then you can very easily print it on poster-sized paper, so you can take the poster home and look at it at your leisure.
It's ingenious.
It's phenomenal. I also used the poster service to have them created with ASM, so I didn't have to lug a poster on the plane. I could just come here and pick the poster up and put it up, so it was very convenient.
So you sent electronically
Correct.
your poster, which is one of those 4x8 sheets.
Yes.
You came here and it was printed here so that we didn't have to see you on the airplane with the huge tube.
I could bring extra shoes.
Beautiful. So how many of these abstracts do you plan to print out?
Well, I actually went through the book and circled some that I'm interested in, and I think hopefully next year, a lot of them aren't in here. Some of them aren't in here, I think, because you had to submit them by a certain date. So I'm hoping that I'll catch on with poster presenters, that it's really to their advantage to electronically submit them so that they can have them available for people to print. Yeah.
Thanks for talking with us.
Welcome.
You can go back to work now.
Okay, thank you.
So that's the new way of printing out a summary of any poster you visit. And in addition, what she mentioned was that you can print your entire poster out here. So in the past, whenever we went to a scientific meeting, you would see the individuals on the airplane with the long cardboard tubes. They were lugging their posters to the meeting. Now you don't have to do it anymore. So another convenience you can find down here on the exhibitors floor. So Samantha, I notice you have a long cardboard tube here, and you're walking away from the poster session. What have you done with that?
I've had my poster on display today. I was presenting it between about 11:30 and 1:30, just showing my results and talking people through what my findings were.
Did you get a lot of visitors?
I did, yeah. There was a lot of interest, and it's linked to one of the exhibitions here because the machine I used was a Bruker Molditoff, so there were a lot of people that went away to go and find out more information from them.
And what was on your poster in general?
It was research using the Molditoff to diagnose cystic fibrosis lung respiratory isolates.
Did you know you can have these printed out here at the meeting so that you don't have to bring them on the airplane?
Yeah, I did know that.
But you didn't do that this time, right?
I didn't know. I didn't get it in time, so I had the fun job of checking it in and bringing it on the plane.
Maybe next time.
Probably, yeah.
Where are you from?
I'm from London, England.
Thanks for talking with us.
Thank you very much.
So that was a brief look at the exhibitor floor here at the 50th ICAAC in Boston. Let's head on upstairs now, where we'll have a meet-up with Frederick Hayden, an expert on antivirals against influenza. I'm speaking with Professor Frederick Hayden of the University of Virginia, who at this meeting gave a great summary of the status of anti-influenza virus antivirals. So I thought we could chat about it a bit. So thanks for sitting down with me.
Great pleasure to be here.
I heard you speak in Switzerland about this 2 years ago, and that's one of the reasons we wanted to have you back at this meeting as well.
Oh, thank you.
So the arsenal of antivirals against influenza is relatively small compared to, say, HIV. Do we know why that is?
Well, it's certainly growing. Historically, we've had two classes of antiviral drugs, starting with the mantidine and then subsequently rimantidine and the M2 inhibitor group. And then in the 1990s saw the development of the neuraminidase inhibitors, which came into clinical practice in 1999. But the reality is that our options are relatively limited currently, and I'm not sure all the reasons why there hasn't been greater interest in developing influenza antivirals, but I suspect it's multiple factors, perhaps including the lack of perceived commercial opportunity, perhaps the false assumption that influenza isn't a treatable condition, and that there's really not the need to provide specific therapy for influenza patients, particularly those with underlying conditions or who have severe or progressive illness.
I wondered if it had anything to do with the fact that there's a very good influenza vaccine, and that is preferable to be used than antivirals in most cases, right?
Well, certainly prevention is our primary means of trying to mitigate the impact of influenza, and it has been for many decades. But really, antivirals are a complementary approach to vaccines, because recall that, of course, not everybody gets immunized currently. Right. There's some seasons when there's a relatively poor match, and the vaccines are incomplete protective, particularly in some of our high-risk populations, so that there's certainly a complementary role for antiviral treatment, and in some cases even antiviral prophylaxis in very high-risk patients who may not respond to our current vaccines.
So you mentioned that the arsenal is growing. So we have, as you mentioned, two targets at the moment for intervention, the ion channel and the neuraminidase. So what are some of the new targets?
Well, the new targets include the viral polymerase, also the virus interaction with the host cell surface receptor. So there's actually an interesting molecule that's a sialidase, which destroys the receptor recognized by viral hemagglutinin. Now, those two targets are still in investigational development in terms of the antiviral drugs, but they're in fact in mid-level clinical development currently, and wait with interest really to see the results of the phase 2 and in one case phase 3 studies that are ongoing with those specific antiviral drugs to see whether they in fact will ultimately prove effective as single therapies. The other thing that's important about these kinds of agents with novel mechanisms, of course, is that they're active against viruses that are resistant to our current classes of antiviral drugs, and so that they would provide either an alternative therapy or one which could be used in combinations to try to either enhance antiviral effect or prevent resistance emergence. But all of this, of course, remains to be determined.
If we did have multiple targets and drugs approved and available to treat influenza, would it be more routine to use a combination of drugs to avoid resistance, as we do with HIV?
Well, it certainly makes sense in certain patient populations. We know from a lot of experience now, including that accrued during the recent pandemic of H1N1, that certainly our immunosuppressed patient populations are at high risk for having emergence of drug-resistant variants. And in some studies, as many as half of highly immunocompromised patients had emergence of oseltamivir resistance when they were treated for their acute influenza illness. So using combinations in those kinds of patient populations, also in trying to rapidly control viral replication in those who have more severe illness or who have high-risk underlying conditions would make a lot of sense. And there have been, in fact, preclinical studies that have gone on for over 3 decades with regard to various combinations of our older drugs, which include the ones like the M2 inhibitors, ribavirin, which is still investigational as an anti-influenza drug in this country, and more recently the neuraminidase inhibitors. And some of these are quite promising in terms of their activity either in vitro or in animal models to show synergistic activity in some circumstances, but there's been really a very limited amount of human testing so far. The one study that has been completed to date was a small one where we compared the activity of the M2 inhibitor rimantadine with a saline placebo to inhaled, or in this case nebulized zanamivir with rimantadine in a hospitalized patient cohort. And although the study was under-enrolled, it did suggest that, in fact, the combination could reduce the likelihood of resistance emergence to the M2 inhibitor, consistent with other work that comes from other antiviral fields as well. So that I'm hoping as we look forward that there will be the opportunity to look at combination therapy, particularly immunocompromised hosts and in hospitalized patients. But in terms of the recent developments, the most exciting news really comes from the study of the intravenous neuraminidase inhibitors. And these agents we know can provide high levels of the drug in a reliable, rapid fashion. And it's hoped that that would then translate into greater antiviral effects and potentially greater clinical benefits, particularly again in more seriously ill patients. And those studies are ongoing at present with both intravenous peramivir, which was recently approved, by the way, in both Japan and South Korea, and intravenous zanamivir. And there are also some studies with intravenous oseltamivir to really fill out the full range of neuraminidase inhibitors.
So these are for particularly ill individuals who are hospitalized?
Well,
right?
in fact, in Japan, the data that have been accrued with intravenous peramivir have been done initially in outpatients, where they looked at single doses given intravenously compared to, in the first instance, placebo, where it was clearly superior, and then subsequently to a standard 5-day course of oseltamivir, where they looked comparable. So that that's a possibility, although I think that really, from my perspective, I see these drugs as being particularly important for our high-risk patients and those, again, who have more serious illness coming to hospital.
Routine administration by IV is probably not going to be very popular in the U.S. anyway.
Well, especially when we have alternatives that can either be given orally or by inhalation. And there's another investigational neuraminidase inhibitor called lananamivir, which has, again, been recently approved in Japan, which appears to have a long duration of activity in the respiratory tract. And so, again, that agent given at a single inhalation in patients coming in with uncomplicated flu looked comparable to a 5-day regimen of oseltamivir, at least in the studies that have been presented to date. So that there's further work going on with that agent as well. So it looks that we will have, assuming that these drugs progress and ultimately become approved by the regulatory authorities here in the United States and in Europe, that we'll have some interesting alternatives looking forward.
What's the timeline? A few years, then we should know the results of these?
I'm really not sure about that. I know that the phase 3 studies of peramivir are ongoing right now. They're about ready to start with IV zanamivir, and I believe that they're expected to be launched soon with the inhaled lananimivir neuraminidase inhibitor that I mentioned.
Uncomplicated influenza in a healthy individual, typically though, one drug, 4 or 5 day regimen is the routine. There's no need to give combinations to those individuals.
Well, in fact, for low-risk individuals who have uncomplicated disease, it's not essential to give them antivirals. It's really based on the discretion of the clinician and the assessment of the severity of that illness, because again, the registrational trials specifically focused on patients with febrile influenza illness. Yeah. So it makes no sense to treat someone who has a mild influenzal syndrome because it's not clear that they would benefit. But we do know now, with further data that's been accrued during the pandemic, that early treatment that's within the first day or two of symptom onset does have benefits with regard to reducing virus replication and reducing symptoms, perhaps reducing the likelihood of transmission of that virus to close contacts, although the results there are not definitive. And it also appears to reduce the likelihood of complications. And there's one study that's in press right now from China where they found a significant reduction with early treatment on the likelihood of getting radiographically documented pneumonia. So that these kinds of data seem to confirm some earlier findings that we saw in seasonal influenza and support the concept that when someone is seeking medical care with febrile influenza, and they can be treated early, and there's sufficient drug availability that treatment can benefit them.
It seemed like last year during the pandemic emergence of novel H1N1 that there was more oseltamivir use than you would normally see seasonally, perhaps because there was some fear of the virulence of the virus. You think that's correct?
Well, there was, to my knowledge, certainly increased use globally of the neuraminidase inhibitors, particularly oseltamivir, and part of this may have been related to the uncertainty regarding the severity of the infection early on. Because as you recall, the initial reports emanating from Mexico certainly suggest that there was a substantial risk of severe disease in these virulent viral pneumonias leading to death in previously healthy young adults in some unfortunate circumstances. And we now have some information about global use, but it concentrated, as it has historically, in Japan, which has had the highest per capita use of the neuraminidase inhibitors in the world, and in the United States. The WHO actually released its stockpiles of oseltamivir for use in less resourced countries, and we're still waiting to see the data accrue with regard to really how extensive drug use was globally, but it did seem to go up in the pandemic context. And I think it made a real difference with regard to the severity of illness in many patients who in fact did have access to early treatment, and more importantly, there's evidence that has now been accumulated to suggest that even somewhat delayed treatment in those who coming into hospital made a difference in terms of their likelihood of progressing to more severe outcomes, such as ICU admission or mortality.
I have one more question because I know you want to hear the ICAAC lecture, and that is the seasonal H1N1 strains eventually became globally oseltamivir resistant, and the new 2009 H1N1 is not there, but it looks like eventually it will also become resistant. If that's the case, what do we do?
Well, I'm not sure your premise is correct that it will become resistant at a global level. To date, we've seen primarily sporadic, geographically dispersed resistance detections, and in the number really, in terms of the total number of isolates that have been screened for resistance to oseltamivir is small. It's less than 2%. Even in a country like Japan, which had quite extensive oseltamivir use, and interesting also, the lowest associated mortality during the pandemic of countries that recorded those statistics. had only about 1% of over 6,000 community isolates that had oseltamivir resistance. Now, having said that, I don't think we should be too reassured that we might not see the same phenomenon as we saw with seasonal H1N1, because when some of these resistant viruses have been looked at in the laboratory, they appear fully replication competent and transmissible in laboratory models of influenza. And we've also seen instances, certainly in high-risk patient populations, of protracted replication in immunocompromised hosts, even when the drug has been taken away, to suggest that the viruses are quite fit. They can certainly cause severe disease and be associated with fatal outcomes in some of these individuals. And importantly, we've also seen then several clusters now of resistant variants being transmitted, including one in the community. So I think it clearly says that even though we haven't seen widespread or sustained community transmission to date, that we can't be reassured that it won't happen in the future, and we certainly need to be vigilant about that possibility going forward.
We'll see. It's hard to predict viral evolution.
Indeed.
Thanks for talking with me. Appreciate your time.
Great pleasure.
Let's go outside now and meet up with retrovirologist Myra McClure and talk about XMRV. We're outside the Boston Convention Center, where you can see the city in the background, and I've corralled Professor Myra McClure from Imperial College London. Thanks for coming outside here to talk with me. You've just given a talk about XMRV, the new human retrovirus, and I was hoping you could clarify it, or the issues around it, for us. This was a virus originally identified in prostate tumors, and since then has been identified initially in some chronic fatigue syndrome patients, but in other studies not. So can you briefly summarize the pluses and minuses and give us your understanding of that?
Of the chronic fatigue story?
Yeah. Well, let's start with chronic fatigue syndrome. Sure.
Okay, well, we are not chronic fatigue syndrome specialists, nor do we have a research interest in that. But we got involved in this story because we had started to develop assays to look at the prevalence of XMRV in prostate cancer, because that's where the virus was discovered. Bob Silverman's group had discovered it a few years previously, and Ila Singh's group had then shown that it wasn't dependent on a genetic malfunction of the RNA gene, but was found in all forms of prostate cancer. So it looked to us as if XMRV might be becoming a big player in prostate cancer. So we immediately thought, is this virus to be found in prostate cancer in the UK? And we developed these assays despite the fact that there had been two groups in Ireland and one in Germany who had failed to find XMRV in prostate cancer patients of their own. But there was one, Nicole Fischer, who had found at least one sample, very low prevalence in Germany. So armed with these assays that we produced, and having got our ethics permission to look at prostate cancer samples that we had in store in our hospital, we started to look for these. And then the science paper was published. And the man who was unknown to me, and in fact I still haven't met him yet, a man called Simon Wesley, who is Professor of Psychiatry at King's College, a neighbouring institution in London, had emailed our Director of Research for our faculty and said, have you seen this paper? And do you have the tools to check out my patients? And this guy, Jonathan Weber, said, well, I don't know much about this virus, but Myra's been fiddling with it, as he put it, in the laboratory, and she would probably have the tools to help you. So we did. We took DNA from patients that he had, 186 of them, and we went into this small collaboration, which only took a few weeks, with the intention of corroborating the science paper's findings, because that would have revolutionized the way he treated his patients. And to our disappointment, of course, we didn't find the virus. And then there was a lot of publicity surrounding the fact that we couldn't corroborate the science paper. And then there followed several other papers, one from the National Institute of Medical Research in Mill Hill, a good laboratory, who confirmed our own findings, that they couldn't find it in their own chronic fatigue patients. And then there was a third paper from Frank Cooperveld's department in the Netherlands. Again, he couldn't find it in his sample. But the one we all waited for, of course, was the one from CDC, because that was an American group, and you couldn't argue, well, maybe this is an American phenomenon and they just don't have the virus in England. So the CDC paper came out, and again, they couldn't find it. So we rather felt that the onus is on the science people to prove that they can find it rather than the rest of us who can't. Does that help clarify where we are at the minute?
You're using PCR to detect the
We're using PCR initially. It was proviral PCR because we were given the DNA, so we couldn't attempt to isolate or anything of the kind. But since we published, we've done some serological assays. Now, these are not assays which are specific for XMRV, because at the time we didn't have the recombinant proteins to do that. But we sent our serum to a man called Dr. Shozo Izui in Geneva, who works on xenotropic murine leukemia viruses. He had some ELISAs going, and he had 2 such assays. We sent them out blinded to him and included some normal controls, and again he wasn't able to find any positives. Of course, there was a weakness in our whole assay because we didn't have a human positive to act as our positive control. So we used mouse serum.
In fact, no study has had a known positive human control yet.
Except the original.
Except the original one. And that would probably help everyone if that were part of the study.
Of course it would.
Even the most recent study, which I'm sure you know of, the ALTER study, which was published very recently, and which did find proviral DNA and some viral RNA in blood, they didn't have a known positive control as well.
They didn't have a known positive control. It's important, I think, to point out that they haven't found the same virus either.
Right.
They have found another member of the family of endogenous retroviruses. Theirs is a polytropic as opposed to a xenotropic virus.
So what does that mean, that humans can be infected readily with mouse virus?
Well, it's funnily, you know, up until this point, gamma retroviruses, which is the genus to which XMRV belongs, have never been known to infect human populations. They're a group of animal viruses, some of which cause leukemia in their natural hosts. But this is the first time that a gamma retrovirus has been connected not just with one human disease, but with 2. So that in itself is quite interesting. I suppose it remains to be seen whether we all are harbouring murine leukemia virus sequences in our genome, or whether it's a subpopulation for some reason or other.
A number of months ago, Steve Goff told me that years ago, when investigators transplanted human tumors into mice, they often became infected.
Absolutely.
So that showed that human cells, at least, could be infected with
humans.
these viruses.
But the corollary of that is that when people were doing things like cancer research in the 70s, and they put human cell lines into nude mice, immunodeficient mice, or human tumors, most mice harbour XMRV, xenotropic murine leukemia virus infection. So the tumors and the cells picked up this infection. Then when they were propagated in the laboratory again, of course, people thought they were isolating a new human virus which was associated to whatever cancer they were looking at. But in fact it wasn't, it was just the mouse cell. So, I mean, retrovirologists, as you know, have got to be very careful. The other source of problematic source of possible contamination are hybridomas. They secrete not only monoclonal antibodies, but you know, xenotropic murine leukemia viruses. So even if you're not using them directly in the lab, you might be using a reagent which has been purified using a monoclonal. So you've got to be really stringent to make sure that you're not just picking up an old friend.
Now, one of the statements in the ALTER paper that I didn't understand, maybe you can explain. They looked at the glycogag sequence of the ALTER virus versus the original XMRV. And they said there was 100% sequence identity with a virus from a lab strain.
I know. This glycogag is a glycoprotein. It's the carbohydrate on the GAG protein. And it seems to have some sort of impact on the pathogenesis of MLV. I didn't quite understand what the relevance was in that paper to glycogag, but there was sequence homology to...
Yes, so the virus, both the polytropic that ALTER detected as well as the original XMRV,
that's right.
They both
have this.
And the Alter virus has a 9 base deletion and the XMRV has 24.
That's right. It's certainly not XMRV. I mean, it very
Yes.
much belongs to
Yes.
the polytropic family. And that in itself is a little bizarre. I mean, why would 2 sets of patients in different parts of America segregate according to what assays that you're using?
Sure.
I mean, that's a little strange.
But it seems that now in both cases, there seems to be this virus in a certain fraction of people, even in healthy people.
Even in healthy people.
So where might it have come from? Do you think it's from a laboratory? I know that it's always possible that it could be contamination, but if it represents a real infection, could it have come from a lab mouse?
If it represents a real infection, it could have come from a lab mouse. It's unlikely, I think, to have come from a mouse to a human. It's more likely, recently, it's more likely to have come from another human who has somehow been infected by mouse sequences previously.
Right.
But you know, we've lived for thousands of years very close to our mice. Maybe the surprising thing is that we haven't found mouse sequences before. We've always just assumed that there are mechanisms whereby we keep us infection-free from murine leukemia viruses. And actually that's something I still don't understand. There are innate mechanisms which are at play that have been overruled to allow us to be, to be infected by these viruses. So that's a little bit odd.
So it could be that a certain fraction of humans have these murine viruses. They're fine, but under certain conditions, chronic fatigue syndrome, prostate cancer, they replicate unchecked.
In the prostate cancers, it does seem to be that this is a very real infection, for the reason that when they've taken XMRV out and grown it in the lab in human cells, the integration sequences flanking the integration of the virus into the human genome are human sequences and not mouse.
Right.
I mean, I think until the same experiments have been done with chronic fatigue patients, you can't really say that this is a real infection.
So in Alter's paper, he suggested that perhaps the fact that the glycogag is slightly different might have influenced the PCR studies in other labs. Do you have any plans to go back and use primers similar
Well, we
to
don't
this?
have to, because when we set up our PCR, we did it with 2 primer sets. One which was specific for XMRV, and that encompassed the 24 base pairs deletion that you were talking about that's specific for XMRV. And another set which we reckoned would pick up or amplify all known MLVs from a very highly conserved region, which was in the Paul gene, reverse transcriptase. So had that polytropic virus been there, we should have picked that up. And the CDC, likewise, they used MLV generic primers, and they didn't pick it up either. So, you know, there's no point in me going back. I just get the same result.
You have no plans to revisit those samples at the moment?
No, I have no plans to revisit those samples because I'm quite confident of the result and the result that we would get. What I have plans for doing, at least I emailed my collaborator this week after the XMRV meeting, is to say, look, we've never looked at fresh samples, and the argument which Judy Mikovits would put forward is, you've all looked by PCR and serology, but you haven't tried to grow the virus in culture. Right. Now, most retrovirologists would say, all right, we'll do a quick PCR first on our tissue, and if it's positive, then we'll try and go back to the patient, get fresh tissue, and we'll grow out. And that's what we're doing with our prostate cancers. We get fresh biopsies, we do a quick PCR to see if it's positive for XMRV, and if it is, then we would go back and try and isolate virus. But we haven't had any positives. Nevertheless, in the interest of science, I've asked if we can get a few fresh samples, fresh blood samples from chronic fatigue patients, and following all the recommendations which are now coming out from the Whitmore-Peterson, and we're getting the sort of drip-fed little hints of how to do it, then I'm very happy to try, and I'm very happy to say, all right, by this method we can find it, if we can find it. But I have doubts about that.
Do you think in the end it will, and obviously this is a story that needs to go on for some years yet before we understand, in the end, could it be that some chronic fatigue patients and perhaps even some prostate cancer patients have this virus as a causative agent, but others don't?
I think prostate cancer is a whole new ballgame. I don't think the two are running in parallel at the minute. I don't think this is going to take years to do this. I mean, already at the first conference for XMRV, there is real willingness to exchange samples, to have panels of blinded samples, to share these among key laboratories to see what their findings are. I mean, I reckon, and you know, at this conference, the head of the NIH was there, and there is a real determination to find out once and for all if this is a laboratory contaminant or if this is real. And I think we'll know by this time next year with a bit of luck. I would hope so.
I think it's a fascinating story. It seems to be a new human retrovirus, and any new virus in people is always interesting.
Oh, it's fascinating. Yes.
So I look forward to seeing what's going on in all the labs.
So do we.
And thank you for coming outside and talking.
Thank you very much. It was a pleasure.
That'll do it for another episode of TWiV. We hope you enjoyed this inside look at a big scientific meeting. I think it's the first time this has been done in this way, so if you did like it, tell us. And I want to thank Chris Kondayan and Ray Ortega from ASM for helping this happen. Next year, the meeting will be in Chicago, as you can see from the big sign hanging in the lobby. And if you like this enough, we'll go there and do it again. As always, you can listen to TWiV on iTunes, at the Zune Marketplace, or on Stitcher Radio, an app for your smartphone. You can also find us at twiv.tv, at microbeworld.org, promednetwork.com, and sciencepodcasters.com, all sites where you can find high-quality science content. Send us your questions, twiv@twiv.tv. And don't forget, microbes, we need them. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.